Good morning again, and thank you for joining us in this online IBEC seminar. First of all, some practical information. Please make sure to switch off your cameras and mics. At the end of the presentation, you will have the opportunity to make questions. Please write them in the chat, indicating if you don't have a mic. Now, let me introduce you our speaker today, Professor Paul Verschure, Group Leader of the Synthetic, Perceptive, Emotive and Cognitive System Lab at IBEC. Paul has received his MA and PhD in psychology and has worked in neuroscience, cognitive science, robotics and artificial intelligence. His scientific aim is to develop a unified theory of mind and brain and validate it using synthetic methods. He has published over 400 articles in leading journals and conferences and is founder and chairman of the Convergent Science Network Foundation. He chairs the annual Barcelona Cognition, Brain and Technology Summer School. Paul is also founder and chairman of the Future Memory Foundation, which aims at supporting the development of new tools and paradigms for the conservation, presentation and education of the history of Nazi crimes. Last but not least, his commitment to apply his research to quality of life enhancing technologies made him found the spin-off Eodyne Systems, which is commercializing novel science-grounded neurorehabilitation, education, and cultural heritage technologies. Paul, thank you for being here today. The floor is yours.
All right. Well, thank you, Teresa, for your introduction. Welcome, everybody. Good morning. So what we're going to talk about is the voluntary brain in action. If I get my slides back, that is. Here we go. And also how our study of fundamental organizational principles of the brain has brought us to new insights in neurorehabilitation. So the general concept of the work we do at SPECS is one of living machines, where we start to think about ourselves and also artificial systems as machines based on identifiable principles that we can also synthesize. The work I'm going to talk about is performed with a team of researchers. I in that sense really believe that team science is the future. It's not a single person who's going to crack this hard nut of how the brain works. So these are the members of SPECS that all have contributed in different ways to the results we're going to discuss today. And also we're supported by a number of European projects without the support of which the work could not be done. So what is really the core issue of the overview here? What we're going to do first, I want to make a few remarks on the brain and its architecture. Then I would like to zoom in on a very specific and well-known and very old notion of learning called classical conditioning. And I want to use that to illustrate a discovery that we made that shows that the brain is actually not necessarily only learning about what the world tells it, but its learning is very much driven by its own predictions of that world and the errors that it might encounter, which brings us to this notion of virtualization, that the brain is more operating in a virtualized informational context than one of a physical world. So we have further been testing that idea in collaborations with Hospital Del Mar, with Rodrigo Rocamora and the epileptology unit, where we found also further direct evidence for this notion, that the brain is virtualizing the world. And lastly, we then have transferred also that concept to the clinic, and that expresses my idea about how we have to validate scientific theory. So it's not just about being right with scientific theory, or it's not being the smartest person in the room. The theory must explain something, make testable predictions, but also give you the ability to control. And in that sense, being active in the clinic allows us to control the recovery of patients who have done that successfully, also building on these concepts of virtualization. Okay? And then I will end with talking a little bit about the system we've built and how we now are distributing that to as many patients as possible, and also what the obstacles are in that process. So here's the brain. It's visualized with a system called BrainCube. It's a neuroinformatics platform that we have built at SPECS to allow us to integrate multiple sources of information, multiple sources of brain data. You see here the white matter tracks. Here we see the different cortical, subcortical structures of the brain that all together give a functioning system that make us do whatever we do. And it's believed that the core ingredients of the system emerged about 560 million years ago, when during the Cambrian explosion, all the main body plans and nervous systems, the templates therefore already emerged in a very short period of time. So how does the system work? This is the big riddle that we're after. And why this is important is not only because it's an interesting intellectual challenge, being the most complex device we know in the universe, but it's also, if you want to make any progress in the human condition, we should try to understand the motor that is driving our behaviors and our experience. Otherwise, we might be not really trying to push future developments in the right direction. The brain affords many capabilities, but it of course also defines the limits of what humans can do and cannot do. So understanding those limits will help us to make some progress, I think, in the human condition. So now, to make sense of this complexity, we start from a theoretical framework called the distributed adaptive control. And what is interesting about DAC is that DAC is changing a little bit the perspective on the brain, because we are looking at the brain as a control system. So, and this is a different perspective than if, oh, the brain is computing or the brain is processing information. No, for us, the emphasis sits really at the other end, where we say, no, what the brain really does is control behavior. So that's the core thing that we're trying to understand. And then from that perspective, we are basically proposing that in controlling the behavior, the brain acts like a multi-layered control system from the body itself, which already gives you implicit controllability features in its biomechanics, to, let's say, the brainstem areas for very direct reactive control, midbrain and some cortical areas for what we call adaptive control, and then more frontal cortical areas for executive or cognitive control. And what's interesting about that is that the brain is, if you want, not one thing. The brain is a very dynamic control system that very dynamically is regulating where it's going to sit in these different layers of control and how it merges them together, right? So, for instance, if you think about these brainstem areas of reactive control, like the superior colliculus, the hypothalamus, the sensory gray, evolutionarily very old, very fast, but not very adaptive, right? If we go to the other extreme, the frontal cortex, where you would find your ability to control your behaviors, to, let's say, not perform socially unacceptable acts in public, and so on, that system is highly adaptive, very sophisticated and advanced. It serves your really long-term goals. However, it's not so fast, and it takes time to attune its properties to the world it finds itself in, right? So that means the brain as such, if you want, is managing a trade-off boundary between the robustness with which it has to respond in real time to the world, while on the other hand, it has to also tune the complexity of that world. And it cannot do both at the same time, right? So your brain is dynamically organizing itself along this trade-off boundary. And this is what we would like to inspect also today, to see, okay, what does it really mean if you are managing, on the one hand, the ability to quickly respond to, let's say, threats in the world, while on the other hand, also pursue your scientific career. So let's see what we can make of that. Now, this is the theory in a nutshell, and what any theory should be able to do is to make testable predictions. And from that, it should also be able to explain key phenomena And lastly, it should give you an ability to control, as also mentioned earlier. So here we have a theory, and it might all be wrong. So how do we make that theory mature? What are we going to explain? And how do we going to make testable predictions from that? So for that, let's turn to a very basic phenomenon called classical conditioning, going back to Ivan Pavlov. Ivan Pavlov, working in St. Petersburg in the late 19th century, discovering what he called psychic reflexes, anticipatory responses of animals to stimuli in the world. And that then became the main objective of his studies from then on. And here we see an example of classical conditioning. So in principle, there would be a neutral stimulus, let's say a sound, and the sound would be paired with an air puff. You see the little tube that will blow the air onto the cornea of this subject, and this will trigger eyelid closure, driven by strongly predefined brainstem circuit. So in some sense, defining a direct feedback loop between the stimulus the system receives, which generates, if you want, an error signal, which we also interpret as pain. It automatically triggers a response that tries to minimize this error signal, and in that way closing a simple feedback loop. But now what Ivan Pavlov observed here, if you look at a bit more detail at this phenomenon, so here we see it in case of a rabbit. We have a neutral stimulus, a conditioned stimulus. We have an air puff, the unconditioned stimulus. And here you have a typical experiment. Here we measure the response of the eyelid over different trials. So this is time. We see the tone comes on at the first trial. There's no eyelid closure. Followed by the air puff. We see a strong eyelid closure in response to the air puff or the shock to the orbit. But with time, we start to see this anticipatory response to the tone. Okay, great. This is what we all know and love about Pavlovian or classical conditioning. So we try to understand, already starting about 20 years ago, the core learning circuit behind this phenomenon. And that brings us to a structure called the cerebellum, and then also a guiding theory. And this guiding theory already goes back to Ivan Pavlov himself, where he says, well, you know, if we look at this conditioning context, what really happens is this anticipatory response is replacing the predefined response. So the tone replaces the function of the air puff or the shock. And this is also captured in very dominant theories of motor control that in generally speak in terms of replacement. So we have very simple feedback controllers. They are driven by peripheral error. Due to the error, a response is triggered, but these are robust and fast responses, so they're not very adaptive. And as a result, these theories propose, going back to Ivan Pavlov, we built a predictive model that predicts that a motor action should be executed. And then by a so-called inverse model, it computes, if you want, what that movement should be. Right? And then in that way, we have still our error signal, but now as opposed to driving this feedback controller, it feeds a forward model that via an inverse model triggers the response. So it replaces the anticipatory response as a replacement. That has been now the dominant view for the last 100+ years. So we can measure this, and for that we can go to the cerebellum. The cerebellum is sort of, in this visualization, the green structure here in the back. The cerebellum comprises the majority of the neurons in the brain, estimated to be about 70%. Mainly the granule cells is the single largest population of neurons in the brain. And what we also know, this is taken from a paper from the lab of Chris de Zeeuw in Rotterdam, that in one very specific area of the cerebellum, lobula H6, you will find neurons, principal neurons, that change their responses in relation to the development of the behavior in such a conditioning task. And we can unpack that structure, and what we then observe is a very specific and beautiful circuit of also clear described by Golgi, by Ramon Cajal as a crystalline, if you want, it's also called a crystalline structure because it's a very stereotyped circuit that is repeated throughout the cerebellum, where in principle we have information about the external world, like sounds coming in over what's called the mossy fibers. They converge on the granule cells we mentioned earlier. Granule cells give rise to parallel fibers that cruise over quite a distance through the superficial layer of the cerebellum, making many synapses as they pass along with the principal neurons of the cerebellum called the Purkinje cells. So every parallel fiber makes just one or two, very few synapses contacts with these Purkinje cells. On the other hand, the error signal, the air puff, the shock, and so on, reaches the cerebellum via the inferior olive, which is the brainstem nucleus, which shows low frequency oscillations, also linked to the rhythms of motor action. And this gives rise to the climbing fibers that are very precise, that target very few Purkinje cells, with which they make many synapses. And as Ivan Pavlov already told us, if we want to understand the physiology of conditioning, we have to find these points of convergence of the signals, because that's the site where the engram, the memory, is formed. So that would then be the Purkinje cell, the principal neurons of the cerebellum that project out via the deep cerebellar nuclei and via this nucleus into brainstem nuclei that, for instance, drive the eye blink reflex we saw earlier. Okay, so we have a very clear mapping here of the behavioral components and the different signals involved onto the circuitry of the cerebellum. So what we started to do already for many years is to build detailed models of the circuit where we look at how these conditioned stimulus signals, the sounds, the error signals over the inferior olive get integrated, and what the main regulators of the responses are in that system. And okay, we replicated many behaviors of that, and we also stumbled into a very specific observation that I would like to highlight. One way to test the model, we've also done this with a neuroprosthetic system. So we put our model in silicon. We use that silicon device to directly replace cerebellar circuits to recover eye blink conditioning in rats, and we showed we could do that. So we have every evidence that the overall way we captured these core principles of organization of the cerebellum were reasonable to the extent that we also can replace cerebellar function. And this is actually the first example of a neuroprosthetic system that is round-tripping signals between brain structures. Usually a neuroprosthetic device is more like an interface. This is the first example of really the real-time round-tripping of signals, because our chip calculated when the eye blink should be triggered, and that signal we send back into the deep cerebellar nuclei to actually trigger that eye blink. But then we put it on the robot to understand, okay, but how would this model really behave under real-world conditions? Because the problem is, of course, if you try to understand these circuits, we are abstracting, we are controlling, and in that you also move away from the realistic real-world conditions in which these models operate. So we have the robot version of classical conditioning. So the robot is in this alleyway. The walls are here red. The green bars you saw are like sound. The collision is like the shock or the air puff. And the robot has to learn to anticipate this collision and then veer away, which it also is able to do. As we see here, it detects the conditioned stimulus. We have the acquired response, and here in the red, we have the response that would be triggered by the collision. So far, so good. However, then we had an observation that bothered us, because actually, so you saw this graph earlier in the video. Here in blue, we see the response to the very first trial. So here we have a collision. In black, you see the intermediate trials, and in red, you see the late trials. If we would follow Ivan Pavlov, we would expect to see replacement. But as you can see here in both the intermediate and the late trials, we still see signatures in our model of both the innate predefined response and the acquired response. Now that was a critical issue because either the model was completely wrong and not really capturing how the cerebellum learns the conditioned response, or maybe Ivan Pavlov's interpretation that it has become the dominant view on motor learning is incomplete. So that now became our quest. Is really the unconditioned response being replaced or not? So for that, together with the Giovanni Maffei and Diogo Pata and other people in specs, we then set Ivan Herreras, we set up actually a comparison case. On the one hand, we took the standard model of motor control, as we just looked at it, where we talk about the replacement of the response through so-called feedback error learning. This is the model that also we described earlier. The feedback error instructs the learning of a predicted response by an inverse model. So we literally took that model as advertised in the literature by people like Cavato and others. And here we defined our alternative. And what was exactly our alternative? Well, there's something that's interesting about this way of doing business. It would also mean that the brain, in learning an anticipatory response, is in some sense making a lot of brainstem circuitry obsolete because you replace. And from an evolutionary perspective, where you also have to optimize metabolic resource, that seems a bit odd. So the alternative we propose is that actually you always keep the feedback circuits in the loop, right? So the only thing that your learning now does, it's not to generate a response. What you learn is an error signal. So that means I start to imagine future errors, and I'm using this anticipated error to now drive the feedback controller. So I don't replace it, I'm actually capitalizing on having it. It's just I start to shift and shape my error signal so I can engage the feedback controller earlier. So did that work? So we taken a postural control task. Why postural control? So imagine you have to catch a ball in this way. You have to generate a counter force, not to lose balance. Well, it turns out that if you look at patients with cerebellar lesions, so the structure we just looked at, they actually have great difficulties to anticipate these kinds of impacts and to adjust their posture, okay? Which is illustrated in these traces here. This is muscle activity, where you see here in a control subject an anticipatory motor response, which is absent in patients with cerebellar lesions, right? So this is why the postural task is a good way to test models of the cerebellum. So here go our two models that we just described. We have first a pendulum on a wheel that we have to balance. So it's a very standard robotics balancing task. In the top, you see our model that predicts error signals. At the bottom, you see the standard model. And actually, when you have a cue that is signaling the upcoming impact, in both cases, they work pretty well. So here you see the two models. In red is the classical model, in blue is our model, and they generate roughly the same angular error over time. So far so good. There's no real difference between the models. But now we're going to go to catch trials. So in a catch trial, the anticipatory cue is generated, but there's no impact. Okay, and now we start to see interesting differences, right? In the top you see our model, in the bottom you see feedback error learning, the standard model, and you see that the feedback error learning model starts to show huge error in probe trials. So it has great difficulties with exceptions. And that in itself is very unique or specific to these models that assume that the world is fully predictable. But we know from motor physiology and also actually from our own experience that we can process and handle these kinds of exceptions rather fluently. So here, and we can look at it again, our error prediction model does not suffer in probe trials because the feedback controller is not disabled. So there's actually an erroneous response, but now the feedback error can, the feedback controller can compensate for that. While in the standard model, the Pavlovian model, if you want, you are completely, you lost that ability to deal with exception. So this is where our model performs a lot better. Now this is an alternative. So now we also understand why actually in classical conditioning then, the acquired response should not replace the predefined response, but they're more in a synergistic relation to each other. So this is now a new understanding of both classical conditioning and of motor control that we can pursue, right? So on the one hand, so we also put this on real robots. Here's our perturbator robot built by Alex in our lab, our 3D printing hero. This is one robot beating up another robot, and we replicate the same result. So it's not, it doesn't depend on some idealized simulated world. We can also replicate this in the real world. So now we have a new insight in how classical conditioning and motor control could really work. And now what are the consequences of that? So let's look at predictions. Well, in the overall brain architecture theory that I've advanced, we are basically saying, look, as you climb up in these layers of control, you become more dependent on representations of the world. The world is more an abstraction. You're less dependent on the physical signals you receive from the world. So there's a process of virtualization, which is also very central to my theory of consciousness. What this experiment now shows us that in this virtualization, we're not only representing and re-representing the world as such, we're also representing our expected interaction to that world and its associated errors. Okay, so our forward models, the predictions we make about the world, is not only about the world as such, it's also about how precise we will be in interacting with that world. And we capitalize on that by solving or fine-tuning this trade-off boundary that we talked about earlier between flexible cognitive control, which is slow, and fast, robust feedback control of the brainstem, right? So the counterfactual error signals, theoretically, that's now the concept, would play a role in coordinating the integration between these layers of control. Now, is that true? Well, a first clear indication comes from this review paper by Peter Strick in Boston, where they basically map out tracer injections of the brain, where they inject in the cortex, and then they look at where are the targets of these cortical projections, and vice versa, they also inject in the cerebellum and basal ganglia. And what they show here is that actually the cerebellum is anatomically, so the structure we just looked at to explain classical conditioning, is anatomically very strongly connected to the cortex via the thalamus and some other structures. So this would suggest that the concept we now have advanced, a predicted error interfacing to the motor learning of the cerebellum, is anatomically not implausible. But now we also found, talking to Chris de Zeeuw in Rotterdam, who's one of the leading physiologists of the cerebellum, he had a puzzling phenomenon to explain. So Chris is already investigating this recurrent interaction between the cerebellum and the frontal cortex for the last 20 years. And what he observed, if we now do idling conditioning, as we just looked at it in the mouse, he saw that actually in the fully trained animal, he sees two peaks of error responses. So he sees the predefined peak not disappearing fully, but also there is an earlier peak before the actual noxious stimulus will be applied. So exactly reflecting what we observed in our robot experiments. And for Chris, this was really an issue. This kept him up at night because it made no sense. This is not a standard model, which is one of replacement. But now we understand that actually this recurrent anatomical circuit is really actively using the notion of error prediction to fine-tune the responses of the animal, right? So we gave an explanation of classical conditioning. We made predictions, and these predictions have been confirmed now also physiologically. So this is great news. But we went a step further, so basically what this means is that the frontal cortex, which is densely interconnected over these fiber tracks also here with the cerebellum, the structure we look at via the pons, actually is directly sending error signals, predicted error signals, back into this motor control system. Now, in parallel, we posed a question about virtualization. And in our experiments with intracranial patients, we then looked at this issue in a bit more detail. This is one of our patients. This is Diogo Pata in the lab talking with that patient. These are different representations of this patient's brain and the placement of the electrodes. And in these experiments, we tried to unpack this issue of virtualization more precisely. And in particular, posing the question, okay, if we talk about memory processing in the brain, do we see any form of virtualization? Do we see any signature that virtualization happens? These experiments performed by Daniel Pacheco. And basically, we placed the subject in a labyrinth. They're asked to go to the red spheres. And as soon as they are at the red sphere, they're in a room, they look at the picture. And they have to remember that picture in that room because we're going to ask them about it later. And then in the recall test, to see how well their memory is, we place them back in different rooms. This might be a room they've never been in before or a room that they have been in before. And we place an image in that room and we just ask them, have you seen this before? And this, of course, allows us to dissociate space, the locations, from the specific item or the picture that they see. So Daniel here used a technique called restatement analysis. I will not go into too much detail on that. If you're interested, just contact me. But in restatement analysis, basically you're considering the brain as a huge volume of oscillators. And these oscillators resonate at different frequencies in a coordinated or not coordinated way. And you use these oscillations, these vectors, these oscillatory responses or their power spectrum as a feature vector that describes the state of the brain. Where you say, at this point in time, I see this distribution of frequencies. And so now you can look at the different contacts you have in the brain with these depth electrodes that we have in the brain of our epilepsy patients and just say, okay, are these oscillatory patterns the same when I'm storing or encoding a stimulus in a certain location versus when I'm recalling that stimulus, right? So metaphorically, you can think about the brain as a choir singing a song. You extract the frequencies in those songs and you say, okay, are these frequency patterns the same or different? And what Daniel found, which was extremely interesting for us, so how you should interpret these plots is this is a correlation plot, essentially, where we look at the correlation between these frequency vectors, right? These feature vectors describing a state of the brain at a point in time. So this is when we present the stimulus at 0, and then we have here 3 seconds after stimulus presentation, and we look here, how similar is the song that the brain is singing at encoding and retrieval. And what we see here is that we have a song or a set of frequencies evolving over a period of about 3 seconds during encoding that is replayed in a 500 millisecond time window when you have to retrieve that pattern. Okay, so in this subregion here, we indicate the statistically significant correlations. And what this shows, which we found very exciting, is that this gives us a clear signature of this virtualization, because as I store a pattern in the brain that takes me 3 seconds, so it's a song that is running over 3 seconds, I replay that whole song, I shift it in time and I compress it into a time window of about 500 milliseconds. So that means this pattern that is now being recreated cannot be dependent on the same analog inputs that the brain is exposed to. So this is an example of this kind of virtualization that we might see in mnemonic or memory processes in the brain. So there's lots of other stuff we can look at here, but I will skip over that. Here we use our BrainCube platform to look at the distribution of the frequency responses about the whole brain. And the bottom line is shown in this animation. All the black dots are all the contacts we had in the brains of the dozen patients that we looked at. And what we highlight here is basically the process of memory formation and also the dynamics underlying this virtualization. That here at the top left corner, you look at the dynamics of the brain as measured with this restatement analysis, so looking at these frequency distributions, where actually when you learn such an item in a location, activity in this structure here, that's called the hippocampus, and the outer shell, called the cortex, is strongly correlated. It occurs at the same time. However, in retrieval of that memory pattern, what we see is that this little banana here of the hippocampus becomes active much earlier, driving, or if you want, coordinating the recall process in the cortex. Okay, so it's really, it has, if you want, decoupled the representation of this item in this location from its physical appearance and the signals derived from that to a fully internally generated representation that is merely triggered by the external event. Okay, so we've now established this notion of virtualization, both of error and of how this also plays a role in mnemonic processes. And now we want to do the last step of a theory. We want to control something. And for that, we go to the clinic. We're going to control a brain that is recovering from a stroke. Now, stroke is, if you want, the simplest deficit of the brain because it's just a hole in it. It's not a very diffuse problem. It's a very localized issue. It can be a big lesion, like in this example. So we just want, and we took stroke because if you look at the impact in society, stroke is the generator of the largest number of disability-adjusted life years. The world population of chronic stroke patients is about 80 million people. And also here in Spain, Catalonia, we have thousands of people a year that suffer from strokes. And essentially, we're not very effective in helping them to recover. Now, stroke is not only that you have hemiparesis and you lose motor control, as we often see in patients, but actually it's a very complex dynamical picture of symptoms that occur in many different modalities and also that occur over a rather broad range of time. So you might start with hemiparesis, motor deficits, but with time we might start to see spasticity, pain, or other associated executive dysfunction, for instance, or depression. So it's not a simple story. But this is the one we now try to attack from this perspective of virtualization. We have built a rehabilitation gaming system, which started about 20 years ago to bring theoretical neuroscience to the clinic in a way to test our theories of the brain. And essentially what we do is we map the physical movements of the patient to movements of a virtual body in a virtual task, because now we can fully control both movement and properties of the task. So, okay, we skipped the video. We have published a meta-analysis of all our studies. This is over 250 patients, more or less, where we show that we have impact on upper extremity recovery, in this case, from 3 weeks to 3 years post-stroke. So this is a very effective method. But I want to zoom in on the issue of counterfactual error, right? The virtualization of error. And we're going to look in particular at experiments that Belen Rubio performed in the lab, where we look at a simple reaching task. Now, a stroke patient will start to show over time something that's called acquired non-use. So basically means if you ask the patient to reach for a target, you will start to see there's a bias. Like in this group of chronic patients, about a year post-stroke, you see that there's a 6-degree bias in bimanual tasks, reaching tasks, where the healthy arm starts to slowly invade or infiltrate the working space of the paretic hand. Okay, so in some sense it means just what you see with those patients, they start to use this paretic arm less and less, even though biomechanically it's fine, right? But it's an acquired non-use, which is problematic because if you use the arm less, it trains less, and you lose even more. So the standard solution that is used across the world is called constraint-induced movement therapy, and Ed Taub and his colleagues proposed this in the 90s, where they said, look, let's just immobilize this healthy arm. So we force the patient to use their paretic arm. So, okay, that looks like a reasonable intuition. It's also widely used. I mean, many publications on constraint-induced movement therapy, but a recent meta-analysis by, clinical trial by Gert Kwakkel and others in Amsterdam compared actually arm use in patients who performed constraint-induced movement therapy versus patients who got standard care, and the difference was minimal, not significant. Now this is bad news because it's a standard intervention method used across the world, and it's very demanding because patients are often asked to be immobilized for hours on end every day for many months. Okay, so it's a very demanding intervention. We sort of force a patient to be really handicapped or incapable of using both hands. So there's a huge cost for patients, and now we also see it's actually not working very well. So we took a different perspective. We now know about counterfactual error. We know errors are virtualized in the brain. So could we capitalize on that? So what Belen did, here we see Belen demonstrating the scenario that she built. So we did, what we added is what we call intention compatible enhancement, right? So we have physical movements reaching for these objects in virtual reality. There's a certain movement error. I have a target, sort of, you see it linked, indicated here. I have a movement, there's a certain distance between the endpoint of my movement and the target, that's my error. What now was our hypothesis? If we minimize artificially that error, that, in other words, I'm changing the error feedback I'm providing to the brain. Since the brain, hypothetically, would use also virtual error to optimize function, this should be effective. So that was Belen's question. So what happened? Well, it's summarized here. We do two blocks of trials with our patients. These are 12 chronic patients. 300 trials was just to get used to the scenario. So we don't change this error, we don't interfere, people just learn to interact with the protocol. And the second block, we do 100 trials where we apply the intention compatible enhancement. Okay? So here we see the -6 baseline bias in pink, that was on purple, that was indicated earlier. In red, we see the effect after this block of 100 trials, and we see with complete symmetric hand use. And then this is followed after washout with a residual error that's still less than the original one, right? So by manipulating the virtual error feedback to the brain, we can restore bimanual and also symmetric hand use, okay? So the hypothesis of counterfactual error really helped us to control recovery. So with that, we satisfy all the 3 criteria for a scientific theory, which is we explain conditioning, we make testable predictions that were confirmed physiologically, and now we also show we can control interventions in neurorehabilitation. So we're closing a loop here, right? From basic principles of brain organization and basic science to application of this basic science in the clinic, which of course, again, gives us access to data. And that is absolutely fantastic. So we're very happy with that. But now, what's next? Well, what next is that then you get patients who ask you for using these kinds of tools. So in some sense, then you have to make the next step. You cannot have your PhD students serve patients across the world. So we started a spin-off company in 2014 called Eodyne, and Eodyne's task is to distribute these interventions to the world. because, as I told you earlier, just for stroke, it's 80 million worldwide. And these people are not doing well, and their life expectancy in the Western world is about 5 years, which is ridiculously short. So I will not go in details of the health economics of this. We did a study with a reference hospital here in Barcelona, how much money it would save them if we would use our technology in the full patient cycle. And in green, we indicate the reduction in time and risk that we can effectuate given the clinical data we have. We have performed over 40 clinical studies with a lot of data. We know exactly where we have impact on this. And it would mean for a reference hospital in Barcelona, we can save them almost 2 million euros a year just in less time spent in the hospital, just the nights in hospital saving they will have. So we're not even talking about, let's say, reduced transportation and so on, right? So this can have a massive impact in the sustainability of our healthcare systems, which we should take serious, because right now our healthcare systems are just not sustainable. So we published many studies from the cognitive deficits related to stroke to aphasia. Great work by Claudia Quetzuta, Martina Meyer on the cognitive interventions. So we have a broad set of interventions here, also summarized in right now a running project called RGS at Home, where we're bringing out this technology to 100 patients across Europe who will test this in an at-home situation in Sweden, France, and Spain. So
RGS at Home is an integrated technology.
I don't think we will really have time for the video.
Science-based rehabilitation solution.
But okay, this is
journey to recovery after.
It works. So by now, RGS is in use in about 50 centers. We have delivered over 100,000 therapy sessions to patients, more than 7,000 hours of neurorehabilitation, only delivered through fully automated computerized interventions, reaching more than 3,000 patients, and working with many clinical partners. So this is going very, very well. What's the next step here? What's our outlook on that? Well, the future. So what we look at here is a glimpse of a future of digital personalized medicine, right? Because you saw on the one hand, we have theory that informs on principles of intervention, right? And that theory is then mapped onto concrete intervention technology. But and in the end, it closes a pipeline, right? Because the basic principles and data we obtain from patients must actually be transformed into patient-specific models that then are used to really optimize and individualize the interventions we deliver to patients, which of course then again creates new data, right? So you're closing a loop. And this is the loop that we're really pursuing, that we're trying to build. So across different projects we have, like virtual brain, RGS at Home, Rehab, and an ERC POC, or proof of concept project. We try to build the different ingredients of this pipeline, because we see this really as the future of digital healthcare. And digital health, which, as you know, is a very central pillar also in future of European funding, because again, our societies are aware that, and even politicians, that our healthcare systems are not sustainable. And we have to rely more on technology to improve that. So this is one form in which we're going to scale to build these automated pipelines. But in addition, we're scaling now to more deficits. So if you look at this, the DALI table for Europe, stroke is number 4, but number 1 is addiction. So we start with collaboration hospital clinic with Lindau Clinic in Germany to run trials now on testing similar approaches and technologies for assessment and rehabilitation of addiction. We look at dementia and the virtual brain cloud project, Parkinson's disease in EU SNN, etc. So we just go down the DALI table. These are the biggest health challenges our society faces in the domain of brain-related deficits, and we want to attack all of them. And in parallel, we're also bringing this to intensive care units for critical care, because, as you know, very relevant now with COVID-19, a majority of intensive care patients go home with serious cognitive deficits because of oxygenation issues, and 75% will not return to work within 3 years as a result. This is an issue we have to address. Okay, so we're trying to do that. Where are we? So we started with theory, an imagined brain and how it might possibly work, right? And from that theory, we made testable, if you want, interpretations, which we turned to very detailed models that were constrained by our understanding of the brain. And from that came new insights about counterfactual error, which we then also found validated by physiological data that coming from the lab of Chris Zale, for instance. This showed that the brain, that more advanced brain structures are sort of controlling more primitive brain structures by manipulating the error signals that they are exposed to. Right? So then from that concept, we went to the clinic to see, okay, can we optimize interventions that way? And I showed you that we could. And this was the beginning, it's an example of the beginning of how we now really want to scale to science-grounded technology that is serving the sustainability of global health. Now, is that a challenge? Yes, because many people don't get the idea of impact and they think that you build companies to make money. That's not true. It's all about serving society. We have to bring interventions to our patients who are actually really suffering. And for that, we need integrated pipelines. And these are integrated pipelines of scientists, research, technologists, but also public institutions. Public institutions have to wake up and understand what their responsibility and role is in supporting these kinds of efforts and not create obstacles in the way because they believe there might be money to be made. No, there are patients out there, we got to help them. And this is one of our biggest problems. I'll give you an example. You've seen in health economics, massive savings. We are active throughout the world, but where's the cattle and health department? Okay, so these are real issues, and these are issues that scientists don't solve. So we also need systemic change, institutional support to make progress here. I think we have pushed this as far as we can. If we want to make impact also locally, we need help. And if that help is not there, okay, then it's going to happen everywhere else except here. And that would be a real shame. Anyway, so this is how we can bring science to society. We're doing it. We're working hard on this as a team. This is a team science effort. And I hope to work with as many of you as possible to scale this to as many diseases and patient groups as possible. So thank you very much for your attention.